Hey guys, my name is Jake Ganley, and I'm a chemistry major here at Northeastern. This year, I've been conducting research in an alternative method for making antibody-drug conjugates. This type of cancer therapy consists of three parts. 1, an antibody that can selectively attach to a cancer cell. 2, a toxic chemotherapeutic agent. And 3, a covalent linker that binds the two other elements. ADCs are known to be an effective treatment for some types of cancer, but there's a problem in the way these substances are manufactured. A traditional antibody has many amino acid residues that the linker can bind to. So when the drug, linker, and antibody react, there's a variability in the number of drugs that can be conjugated to any lone antibody. My research pertains to the way that we make these compounds in the pursuit of narrowing this drug-to-antibody range. My group hopes to solve this problem using flow chemistry. In traditional batch chemistry, whether on a small lab scale or large manufacturing scale, starting materials are stirred in a reaction vessel until they form the product, at which point they are removed and processed further. Flow chemistry is a fundamentally different way of running the same reaction. Instead of stirring in a flask, starting materials flow through and react in a very thin tube, then flow out as your product. The first advantage to flow chemistry is that it's continuous. Because you never have to empty the contents of a reactor, material can theoretically be run non-stop until you run out. Flow reactions are typically cleaner and faster than batch reactions due to superior mixing and heating that occurs in the tube. It is this aspect of flow chemistry specifically that we hope will help improve the drug-to-antibody ratio. Our research has two potentially different yet valuable impacts. Producing antibody-drug conjugates using flow chemistry provides a platform by which a valuable, proven cancer therapy could be produced faster, cheaper, and cleaner. If flow chemistry does in fact lead to a reduced drug-to-antibody ratio, it will have profound impacts on the patients that these therapies are given to. Instead of having to dose this treatment based on a statistical average of how much drug is delivered to the patient, doctors can reliably and precisely know how much drug is in a dose because each antibody will have the same amount of drug on it. Many biopharmaceutical companies are switching over manufacturing operations from batch synthesis to flow synthesis for the reasons that I've already stated, but biologics continue to be made in the same traditional way. While other groups have tried to promote selective conjugation through the use of special chemical reagents, our group is the only one trying a purely methodological approach. Anytime additional reagents are used, there is an environmental penalty, since these reagents themselves do not end up in the product, but are discarded as waste at the end of the process. Flow chemistry is therefore known as a green technique in the field of chemistry, and is an attractive way to produce ADCs. We have proven that we are in fact able to functionalize proteins and commercial antibodies with fluorescent dyes. We've also functionalized a series of 6 therapeutic antibodies with dye under both flow and batch conditions in order to compare the drug-to-antibody ratio under both sets of conditions. Our collaborators at the National Institute of Biotechnology Research and Training in Dublin, Ireland are collecting these analyses for us, and we are anxiously awaiting the results. Our experiments only looked at conjugation through cysteine residues in the antibody. So next, we need to run the same exact experiments conjugating through lysine residues and see if it matches the cysteine data. After we've gathered this data, we need to conjugate actual cancer drugs instead of just fluorescent dyes to the antibodies and then measure the drug to antibody ratio.